In this video, we will see how this molecule, glycogen, is made for storage and to break down to give us energy. So this is glycogen stored in the cells of the liver and skeletal muscles. It's a very branched molecule, and literally branches occur every 10 to 14 glucose units in the form of alpha 1 to 6 glycosidic bonds. Mine is not very accurate, obviously, but just to give you an idea. The blue dots indicate where the branches occur, and the branches form this alpha 1 to 6 bond. There are many non-reducing ends, which are shown in orange in this molecule, but there's only one reducing end, shown in green. So in this video, we will see how this molecule is made for energy storage and broken down to give us energy, or ATP, when we need it. The addition of new glucose molecules, units, occurs at the non-reducing ends only, at the orange portions. And that's also where glycogen is broken down to form glucose 1-phosphate, which we will look at in a moment. But glycogen is broken down at the non-reducing ends. So you can add more glucose monomers to the non-reducing ends, and you can liberate glucose 1-phosphate from the non-reducing ends, not from the reducing end. The reducing end is the part that can react if nothing is bound or attached to it. So it has this OH group that we saw last time on the carbon 1, on the anomeric carbon, which can reduce other molecules. That's why it's called the reducing end. It has this hydroxyl group, which can add hydrogen electrons to reduce other molecules. So it's the reducing end. But you cannot add, that's not where new glucose monomers are added, nor where glucose 1-phosphate is liberated from. That's the non-reducing end. Okay? So now, what I'd like to talk about is how can we actually make glycogen. So glycogen can be produced in a process called glycogenesis and broken down in glycogenolysis. So let's start with glycogenesis. Okay, let me actually bring this, oops, sorry about that. Let's do this here. So we start off with a primer. This is known as glycogenin. We start off with this glycogenin primer. Then we use an enzyme known as glycogen synthase to add the glucose units. However, we need glucose bound to UDP in order to add glucose monomers to the glycogenin and later. So UDP-glucose gives the glucose off to the glycogenin and to the growing chains, forming alpha 1 to 4 bonds as shown here. Okay? As glycogen is branched, right, as you guys already know, there's going to be an enzyme known as amylo alpha 1,4 alpha 1,6 glucosyl transferase. It's a mouthful. Alpha 1,4 alpha 1,6 glucosyl transferase, which creates a branch with an alpha 1 to 6 linkage. That's how you get the glycogen molecule with so many branches. It's this enzyme. So in glycogenesis, you form this alpha 1 to 6 bond. That's where the branches occur. So what I try to illustrate here is that these two monomers at the end are gonna form part of this, are gonna form a branch here. Okay, so that's what I try to illustrate here in terms of formation of glycogen. Okay, now another thing that I'd like to talk about is the other process, glycogenolysis. So in glycogenolysis, we release glucose from glycogen. So just like in glycogenesis, it happens in the cytosol of liver and skeletal muscle. And glycogen is actually broken down into monomers of glucose 1-phosphate, which are then converted into glucose 6-phosphate by an enzyme, phosphoglucomutase. And this glucose 6-phosphate has different fates depending on whether you are in the liver or whether you are in skeletal muscle. So in the liver, glucose 6-phosphate is converted into glucose, and the enzyme is glucose 6-phosphatase. So the liver has this glucose 6-phosphatase which breaks down glucose 6-phosphate to glucose, and that glucose can be sent off to the rest of the body to give it the energy that it needs to and make ATP in glycolysis out of it. In the muscle, the glucose 6-phosphate is used directly in glycolysis to make pyruvate and to generate energy and ATP, okay, for quick bursts of energy. So in the muscle, this glucose 6-phosphate is used directly in glycolysis. Now, I'd like to talk about the process of how this glycogen is actually broken down. So let's look at that here. So the key enzyme is glycogen phosphorylase. Glycogen phosphorylase breaks glycogen to liberate glucose 1-phosphate molecules from glycogen, as is shown here. Okay? When there are 4 glucose units remaining, okay, in a branch, another enzyme, oligo alpha-1,4 glucosyltransferase, another mouthful, this enzyme comes in and literally transfers, it's a glucosyltransferase, it transfers these 3, I drew them in yellow here, these 3 glucose units off a branch to a non-reducing end in glycogen. And then the last glucose monomer, shown in purple, is broken down by alpha-1,6 glucosidase, another mouthful, to release glucose. So this glucose is not in glucose 1-phosphate, it's just standard glucose. The glucose monomers that are liberated from glycogen by glycogen phosphorylase, they're glucose 1-phosphate. But the final glucose here that's broken down by alpha-1,6 glucosidase is just a regular glucose monomer. So most of the glucose that's going to be released are going to be in the form of glucose 1-phosphate that we saw previously. It's only this glucose at the branch that's broken down by alpha-1,6 glucosidase that's just standard glucose, right? And you already saw the fates of glucose 1-phosphate. You know that it can be converted into glucose 6-phosphate by phosphoglucomutase, and then that glucose 6-phosphate can be converted to glucose in the liver to be sent to the rest of the body, or it can be used directly by the muscle in glycolysis to generate ATP. I hope you found this helpful.